welcome back to day 4 of Pediatric Power Up. So just to review where we are, we're nearing the end here. We're on the 4th day of our workshops. This is our second analysis day. So just to kind of recap, we learned on Monday, we learned a lot about different types of data sets. And then on Tuesday, we learned about methods for analyzing those data sets. And then Wednesday, we started to bring them together by pulling data from the Kids First portal and running an analysis using an app, a software tool called DESeq2 in the Cavatica cloud platform. And so today, as again, with our trend of scaling complexity, we are going to go into our Thursday session where we're now not just using an existing app in Cavatica, but we're actually going to build a new software pipeline for a single-cell RNA-seq analysis. So Sangeeta is going to lead this today, and so I will pass it to her here in a moment. And then looking ahead, so tomorrow is going to be a little bit different. It's going to have a different structure. It's going to be more of a conversation. We hope folks will, kind of join a chat and share and talk with us about their individual projects, and we can kind of connect folks with individual resources to try to help, no matter what your project is or what your skill level is, we want to try to help you move forward a little bit further and take it to the next level. So please come back tomorrow. This should be a good conversation. Looking forward to chatting with you all. All right. So with that, I'm going to stop sharing my screen. And so before I pass it to Sangeeta, we have a new face who's joining us today. So I want to introduce Rowan. So Rowan is from Valsera, which is the company that builds Cavatica. And so she is, I guess I can pass you the mic, Rowan, if you want to introduce yourself and tell a little bit more about what you do and your role.
Sure. Hi, everyone. Thanks for the intro. My name is Rowan. I work on Cavatica. So I work as a community engagement manager, meaning that I interact with users, researchers, clinicians. Pretty much if you have a question about the platform, how to access data, like something's just not working right, feel free to reach out to me or contact support, and I'll get tagged in it anyway. So just, get comfortable. I'm happy to respond to you guys via email or whatever you need, and I'm around if you have any questions.
So I was talking yesterday about the blue question mark button on Cavatica, and it goes to the Cavatica team. And so Rowan is one of the people who sees the questions that come in through that mark. So she's hugely helpful, and we're glad to have her join us today in case anything goes off the rails here. But I'm pretty confident. I'm feeling good. We got a great workflow yesterday. And it went smooth sailing, so I'm pretty sure we're optimistic we're gonna get it here today again as well. All right, so with that, I'm gonna pass it now to Sangeeta and let her take us away for day 4.
Absolutely. Welcome everybody. Good morning, and welcome Rowan. Welcome to day 4 of this Pediatric Power Up training, where we're talking about today single-cell RNA-seq analysis and different resources where we can get the single-cell RNA-seq data from. My personal favorite right now within the CFDE umbrella is HubMap. So I'm going to start talking about that. And then I'm going to move on to displaying and demonstrating exactly what I do with that HubMap resource. Let me go ahead and bring up my PowerPoint and then. All right, so as I was saying, today we're going to be talking about single-cell RNA-seq analysis, and I'll start with giving you a brief overview of how that single-cell RNA-seq pipeline differs from what we've learned about yesterday, bulk-seq or like regular RNA-seq analysis pipeline, and then talk about the differences in the resources that we have on Cavatica, like different apps, some of which work on RNA-seq analysis and some of which work on single-cell RNA-seq analysis. And I'm going to show you where we can get that data from, where I was saying that my personal favorite is HubMap, but I'll talk about it a little later. Because apart from getting the HubMap data and everything, we'll also need to talk about setting up some command line interface supported by Cavatica's 7 bridges endpoint set up at the local computer where I'll be working. So I'll show you exactly what steps you need to follow to get that command line interface set up at your end. and then we'll use that data from HubMap, upload it to a project on Cavatica, attach it to RNA-seq analysis pipeline, run through it, and observe the results. That's the basic agenda for today's day. And in the process of doing that, I'll also show you how, if you were to run into any errors, how to debug them. And that's going to be an interesting conversation. I have like both sides of it. I have a successful demonstration that I can share with you, and I'll also show you some failures that I've seen and what methods I employed to deal with how to handle the errors and attach some error handling messages to that. So let's get started. We've talked about RNA-seq analysis yesterday, and David has given us an in-depth description of exactly how that data is extracted and what are the steps to analyze that. So essentially, in terms of the biological understanding that we can extract from a single cell data versus regular RNA-seq data, is that RNA is basically, of course, extracted from the population of cells for RNA-seq data. And then for regular RNA-seq, bulk-seq RNA data, it essentially gives us like a snapshot of the gene expression that is averaged across all cells in a sample. So for example, you have a tissue, and from that tissue, you've extracted cells. And then from those cells, you are extracting the RNA and quantifying that RNA. That gives you average gene expression across all cells in the sample. However, in case of single cell RNA-seq, RNAs from all the cells that are extracted is pooled together before sequencing. And then for the entire population, not just the sample, for the entire population, the RNA sequencing is done, and then that is essentially for the entire cell. In case of bulk-seq, the regular RNA-seq that we do, it does not capture the cellular differences, like at the cellular level, the differences that occur within like a population. So if there's like some conditions that happen in a certain, let's say we're talking about some kidney cells. So there's the kidney medulla and the kidney cortex as well. So within like a population of cells, the differences in the gene expression in those populations, that is not essentially captured in case of bulk-seq RNA-seq. But that is the kind of difference that we see with single-cell RNA-seq. We would be able to individually use isolated cells, and we would be able to process them separately to understand the gene expression profile for all the different subpopulations. That is how we would be able to examine the cellular heterogeneity from using single-cell RNA-seq. And that's why that kind of single-cell RNA-seq analysis methods are very useful in identifying like rare cell types, or basically just understanding what kind of diversity happens within the tissues and characterizing the different cell states and transitions in case of healthy versus non-healthy, or conditions where a patient or a system is undergoing some kind of drug treatment or any sort of external interference, and then understanding what kind of characteristic changes those treatments may be introducing into the gene expression profiles. That's what we study from single-cell RNA-seq. In terms of the data analysis pipelines for both, for example, DE-seq2 and this single-cell RNA-seq pipeline, there are some differences there as well. For the DE-seq2 that we looked at yesterday, it started with like using FASTQ files, and then we generate alignment files. And then essentially the process of moving from a regular sequencing file and then using the counts matrix data, and then using that information for visualization and normalizing the counts data. That is like the general pipeline. You start with sequencing data, you do the like you use sequencing data, you pre-process it to remove any sort of external impacts, like, you know, the adapters that we we're talking about earlier on day 2. So we use FastQC for like alignment part of it, of the pre-processing, and then like alignment, FastQC for the pre-processing and data cleansing part of it, and then star can be used for alignment part, and then feature counts for extracting the accounts information. and then using other methods to normalize that counts data before visualization, before performing any sort of PC or clustering on that data, so that that data can finally be passed through the pipeline, the one that we saw yesterday, DSeq2, so that we understand how differentially these genes are expressed within the population that we're studying. And these pipelines, this general set of steps is different from the single cell RNA-seq, because in case of single cell RNA-seq, we use different types of files. Those are called like Surat objects or single cell RNA-seq objects, and they are data objects which hold different slots of information. And then those slots of information, they are handled differently by the different libraries that we use, such as single cell experiment or Surat or cell ranger, those kind of libraries. And then that format is specific for only those, although we have like regular RDS files also, and RDS files, for example, they can be read using a different like a regular data reader, like read RDS in case of R. But then there's also other kind of Surat objects kind of information that can be stored in such RDS files also, but then they're read differently by the libraries, the packages that work for single cell RNA-seq analysis. That is like a brief introduction of what is different in terms of the biological data capturing process for single cell versus bulk-seq RNA-seq analysis, and how the process of analyzing that data using bioinformatics pipelines differs in terms of the data files, the libraries that we use, the pre-processing steps that we may follow for both the set of analysis, and how we might want to visualize that data. All of that we are going to look at in just a moment, but this is like a general steps of what we're going to walk through using Cavatica and the data resource hub map. I will leave this here and stop sharing my screen, because now we're going to do things more hands. So let's get started. I'll bring my browser window up and then navigate to hub map and share my screen again. Let's start with the hub map consortium, and here's a link to that. Let's go to the home page. Hub map, as you can see here, this is a huge data resource that captures a variety of data sets ranging from RNA-seq analysis to proteomics data, like genomics data, proteomics data, data visualization software, data visualization tools, everything in one place. So this is a very versatile resource to be able to use when we want to conduct an interdisciplinary analysis, where we are working with different tissue samples, where we might want to work with different data types. We can find all of that information in one resource. On the HubMap data resource, if you look here, describes how many types of data sets are available on this resource, based on organs and based on the assay types that were performed on these tissues from various organs. Let's go ahead and explore some of these data sets. Let's start with the data set type. Let's look at, first of all, of course, because we are going to look at data on like a publicly shared resource, let's only look at the public data, not any of the protected data, because anyway, I may or may not have access to that protected data. And even if I had access, it wouldn't be wise to display it with everybody, share it with everybody. So let's only look at the public data. And from that, I'll only look at data that was processed within the hub map, and I will look at single cell kind of assay. So the modality that I want to choose here is single cell data, and then further I could just specify whether the type of data set I want is ATAC-seq or RNA-seq or anything else. Let's look at what options I have under the RNA-seq analysis. I also have access to like bulk RNA-seq data and single-cell RNA-seq data. For now, I will look at the single-cell RNA-seq data, and I see that I have some 43 results found from different studies and from different organs. And this is all published data as we see here. If we look at a certain file, a process data set, we can see that it captures the expression data in raw and processed format. This processed format is going to be in the and data kind of version, and data is the Python package, an equivalent of which is also available in R. And this is a process data file. And we will be looking into that data set in R just a little later. But about HubMap, about this data set, HubMap also offers the visualization tools that are associated with this kind of data set. This is a single cell RNA-seq data, and this is a scatter plot for the that data set to see what kind of genes are available and how they cluster, how they differ from each other, and how they might be similar to each other. It also captures metadata about the donor, metadata about the origin of where that sample came from, and a lot of other files, including the FastQC output. Everything is available here, and there's an option to transfer the HubMap data using different resources such as a Globus access. I'll not be demonstrating that today, but there's an option to do that in multiple ways. So I'll share with you all one way of extracting some of the HubMap data. We will be looking at some sequencing data that is available, that is published on HubMap and is also available on NCBI SRA. And we'll find a way to import this SRA data on Cavatica and process it to extract some FastQ files in a Cavatica project. So let's get started. I am here on the NCBI SRA page, and I will use just HubMap as a keyword to see what kind of data sets I have. So I see that it has a lot of search results, and out of that search results, let's also again only look at the publicly available samples. It has two. I'm looking at the bioproject filter, and out of that we are looking at two different search results. Let's open one of them. It's taking a moment. What we're going to do here is we are going to extract the metadata file from this, and then from that metadata we are going to extract the paired and FastQ files using an app on Cavatica. So this is one of the search results that I've opened here, and I'm going to, okay, I'm looking to essentially I'm trying to find a way to send it to a file of, okay, let's try with the accession number. Now that we have the file, let's search through the SRA using not just the HubMap kind of or the bioproject kind of filter. Let's specifically search for this accession number within SRA and see where it takes us. That's just my way of making sure that I'm able to extract the metadata files from that. I could choose any of these. Let's say I choose this, and then I open it, and I send this information to the run selector to finally extract the metadata. It's going to take just a few seconds. I've seen that sometimes the metadata files are huge. So once we have that metadata file, I'll also edit it a little bit to only include a shorter list of the SRA accessions.
Sangeeta, while it's loading, could you share a URL with folks in the chat just so that way we can follow along a little bit closer? Thank you.
Absolutely. So this is, well, let me go to the NCI SRA homepage. Okay, here's the link to the NCI SRA homepage. And once again, when you browse to the page, search for the word hub map, and then from the word hub map, when you get a list of results, only look at the public projects, because you might or might not have access to the protected version, protected results from that data set. And then from there, from that result set, find the accession number of any random search result and search for that accession number again through the SRA. And then from that, send the results, send the file, send the result to a run selector. And when you run it through the run selector, you'll see a page like this, the one that you have on screen, from where you'll be able to download the metadata. Okay, I think the metadata is downloaded. At mine, it's working. And while this happens, I will also show you what to do with this data set from the hub map, or especially because we are going to, I'm going to show you how to upload these data files, the raw expression and the expression process files for expression data for single cell RNA-seq onto Kavatika's project. But downloading these files takes a lot of time. So if you want to work hands-on with me, maybe you should start downloading any of these data files. Download the expression and the raw expression data files here. I'm not downloading it because I already have them, but you could work with it. I have clicked on download, but I'll stop it because I don't need that. But yes, you might want to start with that. And I have the SRA run table here that I'll open for you. This is what the file looks like. And this is a slightly, this is a decently small file. So I think I've picked a good sample. I'll let this file just the way it is, be just the way it is. And I will upload this. I'll rename this to whatever my accession number was. This one, so that when I upload it on Kavatika, I have just a moment. All right, so I've uploaded, I have renamed that file and I'm going to open Kavatika. We have like a multiple things going on here because we are trying to do as many things as we can within the 2 hours. So on one hand, make sure that you started the download of the expression file and you have the metadata file extracted from the run selector. And then let's go to Kavatika. I will again log in with my ERA Commons ID here. And I will authorize my NIH Gen 3 Data Commons connection to my account. And now that we are here, I'm going to open a new project that we can work with today. I'm going to create a new project. Let's call it Power Up Day 4, Power Up 2 Day 4. And I'm using my pilot funds here, and I will make sure that my spot instances execution settings is checked. And on the advanced settings tab, I will allow for network access. And let's just review the project name here. As you see here, Kavatika in the URL that it creates for each project, it ignores the cases, and the underscores are not accepted. They just use a hyphen. So for convenience, let's not use a underscore in the project name and use a hyphen just to match the URL appropriately. Because we are going to be using the CLI to upload to this project later, this will be more convenient, more intuitive. And let's create a project. Now that we have a project, let's go to the next tab and upload the metadata file that we downloaded from SRA. And as I said, I have renamed the file to include the accession number. Project creation step. Okay, project creation is relatively simple. I'll quickly show you the project creation. This upload is going to be very fast. It's a small file. The file is already uploaded. We are on the dashboard. But for Harry, you can go to the projects tab and look at all the projects that you already may have access to. And I have a lot of other projects that I've been working with different collaborators or just myself. But you can click on this plus sign to create a project to get started. All right, Harry. Now that Harry is also all set, I hope everyone else is as well. And we have a file here. We are going to go to the public apps section. And we are going to use a pre-built Kavataka app, which will use that metadata file and extract the sequence reads attached to that metadata, link to that data, so that it can be used for downstream analysis in whatever kind of pipelines you may want to use. So this is just a demo one, the file upload step. All right, sure. Before I get to that, let's just copy this tool into our project. Harry, before I take you back, make sure that you can also work with this app copying and renaming of the app as you see fit. And copy. This is also going to be very quick and you can navigate back to the project. And for Harry, I'll take just a moment to show you how to upload a file. Go to the Files tab, you add files, and then you have these options of different modalities where you can access your files from. You could access the public files, just like the KF references, Kids First references files that David showed us yesterday, or projects, other projects, files from other projects that you may have access to, or from your computer, or just FTP, just like instead of using the wget that David used yesterday, you could provide the FTP link over here and still get the file. And then this is the DRS, the data repository service that a lot of NIH resources support. And you could just paste the link, the DRS link for that data file you intend to work with. Or you could also work with volumes on AWS or Google Cloud. I'll show you what it looks like, because I have access to one publicly available data set on Kavataka. It's called D3B Open Targets, and I have access to that. So when you click on volume, you'll see a screen like this. I don't want to import any file from this right now. And Harry, hopefully you've been able to upload a file here now. We'll go back to the, you can name it the SRA metadata converter. Okay, I'll show it to you here on this project. The app is called SRA metadata, SRA download and set metadata. Did you get that? When you search for this in the public apps, you should be able to,
Thank you.
Uma. Thank you. Yeah. And thank you, David. We are going to go ahead and run this. And this is going to run pretty quickly because we are working with a small metadata file. So as you see here, I'll go back one step. This was going too fast. We have the SRA download and set metadata app that is available inside the apps tab for us. We can just click on run. When we click on run, it will open up this tab where we can change the name of the task. Let's change it. Power up to demo. Let's call it the power up to demo and attach the file. It will show us the list of files that are available in that current project. We can also access other projects and the files in other projects and some files that are publicly available on Kavataka. But for this app, we are only going to use the file that we just downloaded, the metadata file that we downloaded from NCBI SRA. And we are going to save that selection. This particular app does not require for any of the other app settings to be changed. It can work with the default settings. So we'll just let that be and hit run, and we'll see what the output files are going to be. This might take just like a minute or two. And while that happens, we'll see if you all can go back and see if your data download from the hub map is going well, or how fast it is, or if you have a time estimate on that. If there's no time estimate, that's fine, because I have some files at my end that I have locally, and I also have them available in a different Kavatika project, and we won't be delayed. Let's see what's happening here. It's currently running. It was queued earlier, and now it's running. And if you go to the tasks tab, you will see that you'll see a new task here. It's called the Power Up demo. And because I had earlier switched from the same screen to go back to the run selector, I have a draft that was created here earlier, which I don't need, so I will delete it. And here on the tasks tab, we can keep a tab of what tasks we have running or we have completed, or any other status like aborted or failed or anything at all. And when it's done, we'll also be able to see the cost that incurred in terms of the data storage and for the computational resources it needed to use to process that data through the pipeline. Okay. Because insufficient funds in your billing group. Okay, Harry, did you request for pilot funds using that email address that David had shared with everyone? Rowan, you can reach out to Rowan, send an email to Kavatika Velsera's support email, and you can CC Rowan to help you get started to have the pilot funds in your account.
Thanks, Jane.
All right. And while this happens, I will just show you locally what that data file looks like. Give me just a second. The expression data file looks like. All right, so yesterday I had prepared for a lot of data files to be downloaded locally, because I knew that this would take a long time today, and it would take longer than maybe sometimes a couple of hours, because I was downloading multiple files. But essentially, when you download that file, you will see something like the data file locally here, and you can see that these are like, they're not like very big, but they're not very small either. They're in the megabyte size. And when you work with these kind of files and when you have to upload them to Kavatika, there's an easier way to upload them instead of using the GUI to just click on, files and adding files. Instead of doing this, what we can do is upload to the Kavatika project, the same Kavatika project, using Kavatika SBCLI. If you see here, this task is still running, but in the file section, you will see an additional file. In the files tab, you'll see an additional file, this one. This is a log file that is generated from that metadata table. It's sort of the same size, but that's one of the expected outputs of the task that we're running. It was just a couple of SRA sequence reads. It's taking more than 3 minutes. It ran faster yesterday for me. It just took me like 3 minutes to have a few sequence reads extracted. Maybe these are bigger reads. Let's wait for a second. But in the meantime, I will give you an introduction to the Kavatika CLI, so we can get started with the data upload. To work with Kavatika CLI, you would first need to look at the documentation. One moment, I'll give you the documentation. Yes, if you see in the chat, I've provided a link to a Kavatika documentation, which talks about how to get the Kavatika API set up. What that means essentially is that your local machine needs to talk to the Kavatika backend computer where the tool is deployed, where the platform is deployed, and to that address, upload the data file that you're trying to work with from your machine to the project on the tool. That's what it essentially means. And that's how you need to build a connection between your local machine and that project that you have on your Kavatika profile. So if you read this documentation, you will see that there are a few basic commands that you need to get started with. And you need to work with something called an SV pack and install it. And this is a relatively simple process to get started with. It's as simple as like a pip x install SV pack if you were to do it locally. And I've provided that kind of command in the chat as well. Let me just refresh this page to see if there are updates to this project, to this app, the way it's running. And once you install SV pack locally, you will also need to set up local credentials. What that means is, while you want to upload data or deploy an app on Kavatika, you will need your Kavatika platform account credentials to also work with your locally installed Kavatika API endpoint, essentially. And for that, you need to use credentials that are specific, that are unique for you as a Kavatika user, which you will be able to see under developer. Now, I don't want to show you my authentication token, but you will need something like an authentication token that you can see on your developer tab, authentication token link, and you can set up your local Kavatika credentials using commands like the one that I've entered on the chat. You'll need to create a new file locally called 7bridges credentials. And this file will require for you to name the profile. I've called it Kavatika. Specify the API endpoint, which you will see there. And for that endpoint, each user, each developer has an authentication token, which you can enter here, as you find from your Kavatika home screen. And then once you have that set up, you can go to your command line to upload. Now, where's my command line? Did I open it?
Sorry, we can see the demonstrations you're doing.
Right now, it's only the Kavatika screen. I added the rest of the commands on the chat. Can you see the chat? I added the link to the Kavatika documentation and the... Yes, absolutely. This is a pretty advanced. And while we're waiting for this app to complete running, we're just moving a step ahead. That's why it's a little more complicated. So Rowan is right. Don't get stressed. If you're not able to follow along today, you will receive the recording later. And of course, we will be here. You can reach out to us via email or send us a message via LinkedIn, and we are happy to help you. Rowan is also an amazing person who helps with all sorts of technical questions. So please feel free to send out an email to Velsera team, to Rowan, or to us, David and myself. Yes, okay. So now I'm going to go to my command line. Do I have my command line here? So I'm going to navigate to the file where I have all of my data downloaded earlier. As you see here, I have a bunch of directories and files which I pre-processed and saved here. And out of this, I'm going to upload one file onto the Kavatika project that I'm currently working with. Now, if you have the Kavatika command line, the API set up, then you will be able to work with some basic Kavatika 7bridges commands on the command line, one of which is going to be for an upload. I will bring that up over here. It's just as simple as SB upload start, and then it's followed by the path of the file that you're trying to upload and the destination where you want to upload that to. And the destination is going to be the project. So in here, I will go to the project information, look at the settings to get the complete name of the project. Okay, I know it. I mean, I know what my project name is going to be like, but I was trying to show you where to find that project name in terms. Rowan, do you remember exactly how to navigate to the screen?
If
where
you
you
click on settings, I believe you can get to the project information. Oh, sorry, within the project. So how you can do it here too. You can click that pencil button under project name. There you go.
But not the entire link, like the username, the project
Oh,
name.
the entire link should
Yeah.
be in the URL, I believe. Like right now it shows a shortened version, but you see your username here. And then the name of the project. So it should be, start with the SH and then go up to power up to day 4.
Thank you. So as you
No
see
problem.
here, everybody, this is the proper URL that we want to use, and this is what I'm going to copy onto my command line, onto my terminal. Here, we'll start SB upload start, and the file name. The file name that I'm going to use here is HBM339expr-h5ad. This is the file I'm going to look to upload, and this I want to upload to the destination of the project name that I'm working with, day 4. And it's also possible to add a tag to this, just so that we can tag the file names. Yes, Rowan is right. So that you can identify the file that you're uploading, Power Up demo. And here goes. And this is a lot faster compared to using the GUI to upload files to Kavatika. As you'll see, it'll take just a few seconds. Okay, the upload is done. This is still running, but
Hi,
anyway, while
Sangeeta.
this, yeah, go on.
Just a question so that I follow the rest of it. Are these first 3 steps on a local computer on the command line trying to authenticate? Oh, I see.
Correct.
Thanks.
Yes. Yes, you want to install the SB pack first. And
Okay,
once you've installed the SB pack, you want to initialize the API endpoint by attaching proper credentials. The
Okay.
credentials where you give it a profile name, link the API URL,
Mhm.
and use an authentication token.
And that authentication token is coming from the developer tab authentication
Correct.
token
As you
from your
understand.
account.
Correct.
Okay,
As you
thank you.
No problem. All right, so while this is working, I will go back to the files tab and show you the file that we just uploaded with a tag. And while we have one app that is still running, I'm going to show you another app which is going to work with this data file. It's called this other app converts the single cell data file from one format to another format, single cell object converter. I'm going to copy this file, this app, into the project that we just created. And if you need the name of the app, again, it's single cell object converter on Kavatika. So let's go back to the projects and look at the apps that you are working with, and go to this new app that we just copied into our project and read through some of the documentation. If I remember correctly, it converts from Surad to single cell experiment. Okay, so I want to demonstrate this, but it converts from Surad to single cell experiment. So I'll show you another way to convert the H5AD file format to Surad object. You could also use Data Studio on the Kavatika platform, but I have not created that, and I've not had all the libraries and everything ready on the Kavatika Data Studio. That's why I'll go back to my apps, but I will show you. I will bring up an R screen here. Again, this is a slightly messy file because I was working on it earlier, and I had installed a lot of the packages that I needed to work with, such as Rod disk or TX import or single cell experiment or and data, which is the package that can handle the H5AD file. And I have already invoked the libraries that I want to work with. And I will also read the H5AD file from my local object. Let's call it just the HD5 object, H5AD object. Okay, and I will just run this. And once we have read that data file, the one that we downloaded from HubMap, into R object, we are going to use an R package and convert the format of this H5AD file from ANData to Surat and name it slightly differently. We are going to run this to create a Surat object. Now and I'll also show you what a Surat object looks like. Now this format conversion has been split out into a file, but not into an R object. So let's create another R object which reads this Surat object. It is not reading it because it's compressed. No such value directory. Yeah, that might have been a typo somewhere, but let's read this Surat object. And I'm going to show you what this Surat object looks like by running commands as simple as what this Surat object is. If you see here, the description of this RDS object is that it's an object of Surat class type, and in this Surat class type, you'll see that this data object, this Surat object, is not a simple table. It's not a simple matrix of like an M by N style, but it has a lot of these slots. And it has a lot of these different values inside it, like dimensions, dimension names. The data which would essentially be the counts that we're going to look at, and it will also have the names of the different genes that it's tracking. So basically, this is a different type of data object that is not as simple as just an M by N type of matrix. And that is why the packages that deal with this kind of data are also different from the regular kind of read RDS or read TSV or read CSVs or any other data manipulation functions that we might use for other kind of analysis, like single cell bulk-seq RNA-seq analysis or DE-seq2, which works with simpler data formats. If you see here, we have created this RDS object file, and we are going to use this file to also upload onto our project. We are going to just copy our project and replace the file name here. We are already in that same directory where the file exists, but since I've copied the entire path, I'll just let that be, and I will go ahead and start uploading this data file to the project. Let's see how many seconds it takes, and that was pretty quick. Let's go back to the Kavatka browser and look at the tasks. Have we completed? Yes, we have completed the metadata extraction from the task that we had run earlier. Let's look at the results. If you see the results here, it has extracted a FASTQ file. This is how it works. If the sequence read was a paired end read, we would see multiple FASTQs with like the SRR number _1, _2, _3, something like that, which I can show you in a different project when we come back to it. But let's also start another task where we can convert this single cell object into a Surat object. Let's just use this, run it by attaching this Surat object and let it run to create. Again, here we don't need to change any values for any of the other app settings. And the output of this execution is going to be a single cell experiment, an object of the package type of the class single cell experiment. While this is running, I can go to my projects and my rehearsal, and I can show you a different yes, this is what I was talking about. I had run that SRA download and metadata earlier for a different set of accession numbers. And for those, I was able to get the fast queues extracted. And this was a paired N read. So I was able to get the output for each of those pairs in the fast queue format. I'm showing this because all of this still comes from the hub map, and the associated data from a hub map submission can be in different formats. It could be something like just the reads in the FASTQ format, or an entire single cell object, or some visualizations and everything. So I'm trying to make sure that I cover as many aspects of the versions of data available on hub map. Let's go back to our task, and see if the other task not on this project. See, that's what happens. There are multiple projects, so I lost track of the project I was in here. The 16 minutes of execution that ran earlier to extract the metadata, it might have been a huge file, but it cost us 19 cents. Thankfully, I have some more funds left in my pool of pilot funds, so we'll be fine for today. And this should happen pretty quickly. It happened in like 3 minutes yesterday, under 3 minutes, for me to be able to convert from a Seurat object to a single cell experiment. And the reason we are doing that conversion is, one, we can see how versatile some of the apps on the library of the pre-built apps available on Kavatica is, and then use the output of this, whatever the output of this task is going to be, as an input for the next app that we're going to use. And to get started, let's copy that app. We are going to go to workflows and tools again. And we are going to browse the apps for, again, single cell. And we are going to copy this one, single cell. What was the name of the app? Let me go to my other project and just make sure that I have the correct app here. Multi-sample clustering. Why wouldn't it show up here? Here, we found it. We're going to copy this app into the project, and we're going to also edit the project name, the URL for that project name, and copy it over to our project. Once we have that, we'll go back to our project, and we'll see that now we have multiple apps here that we've copied from public projects, public apps on Kavatica. Failed. Okay, it ran just fine yesterday, but let's see what happens if it fails. We're going to look into the details. And we can do that by going into the error message, like here, show details. And we can also look into the view stats and logs. And when we view logs here, we can see the error message. Argument is of length 0. It's okay. I do have other things that I want to share later on how to exactly debug. But before we get to that, I want to show you how to successfully execute one of these. So I'll just go back to this other project that I'd worked on yesterday. and I'll show you the task and just use exactly the file that I had used yesterday. I'm going to use the Surat RDS file. I will remember this file, which I will copy into the current project and then attach it to the same app again. As you can see, it's also easy to copy files from one project to another. We are going to copy a file from this rehearsal project, which is called Surat RDS. Let's look at the RDS files. It doesn't show me this file here. It's this one, right? Yeah. This is the file that we are going to copy to the current project from the project that I was working on yesterday. I can choose to keep the pre-existing tab or not, and add another tag. And this copy is also going to be pretty quick. And again, the reason we are doing this is that the output of this we are going to attach to the new project that we copied just now. We are going to go back to the current projects failed tab. We are going to edit the parameters, the values, the files that we attach to it, and run it again. What's different between the two? Oh, the file name is slightly different. Okay. Now let's run this. It ran yesterday in about less than 3 minutes. Let's see what happens today. Okay, where is the Surat file coming from? Okay, so when we downloaded, remember we downloaded the H5AD file from HubMap. And then after downloading that file, I used locally an R script, an R command to convert that H5AD file into a file format that is compatible with some of the apps that I'm going to be working with on Kavatika. Now, you could also do this kind of file conversion or like R script, running any sort of R script or Python scripts for data manipulation, even on the Data Studio on the Kavatika platform. But I chose to do it locally because I had all the libraries that I would need preset already on my machine locally. That's why it was faster for me to quickly run that file conversion and also demonstrate how to upload files onto the Kavatika project using command line. That's why I did that. And the next question is, did you have to import that or it came from the SRA? Okay. That H5AD file was downloaded from HubMap, like using this download button here. But downloading this could take up to like 30 minutes or 40 minutes or over an hour sometimes. And if I were to do that right during this training, it would take a very long time. So I figured I'll just get ready and prepared with all the data files I may need for this training for today. I hope that's helpful, Harry. And I understand that today things are getting a lot more complicated than we have done over the last few days, but that was the goal. We were trying to make sure that everybody was comfortable with this level of versatility of features that the Kavatika platform provides, and learn as many aspects of that whole process about downloading data, uploading data onto Kavatika platform, after downloading from various resources, including the different data resource centers under the CFDE program. And once you have them on Kavatika, I wanted to show you as many apps as possible that are publicly available, developed by the Kavatika developers, and use many of those for an advanced analysis that we're trying to run right now for single cell RNA-seq. And that's why we are trying to do like multiple things here. Can I get a web scrap from Kavatika by Selenium or Beautiful Soup? Rowan? I think this is a Rowan question.
Hold on one sec. Let me check it out. That's a great question. We do have an open API. I don't know about web scraping and how that applies because technically this is associated with like NIH and I don't know what government guidelines they have. But let me find out. I will get back to you.
I copied a file that I expected from the successful run of this other app that I was trying to run. So I'll show you what I have done in this other project. Earlier, I had run a lot of tasks and edited some of them, the apps, to see what happens. And then I also ran this file converter, the object converter app that I was demonstrating to you. I uploaded this file from my local machine that I had created from that HubMap H5AD file, used the R script to convert it into an RDS, a Surat object of the RDS extension, and uploaded to my other project, and then ran this app to retrieve this output of a single cell experiment, an object, a data object with the extension RDS, which is of the class single cell experiment. So what I've done now is I've copied this file into the project that we're looking at today. If you see here under the files, I have made a copy of that file during the break time. This is the object. And then what I did was I attached it here to the app for multi-sample clustering and gene marker identification with Surat here as a matrix for the counts, gene cell counts, and ran this. The other settings that are required for this app to work successfully are specification of the ensemble version, which I've specified, of the genome build, which I've specified. This is a human sample, so we're looking at the reference genome of the for humans. So I've specified that, and I've specified the input type. We have something in the chat. Yes, counts for expression patterns. That's right. That's what we have in this. That's what we have in this RDS object. That's what we have in the single cell experiment class RDS object. And we attached it to this app as an input, and we ran it. And if you were to see the other project again, I can show you how long it took yesterday for that task to run successfully. And I ran it like multiple times. I've done multiple edits to everything just to see how I can use that information today to demonstrate to you all what parts of the app in terms of the code can you edit and how to upload it back onto Kavatica, how to deploy it back to Kavatica, and then run it again. So yesterday, the successful completion of the app that we're running right now with the output that was expected of the other single cell object converter app. It took 6 minutes yesterday and it cost me 3 cents. Let's go back to the current project. And once this runs successfully, that's the hope, that's the goal. It ran yesterday fine, so I'm hoping it'll be fine. We have 3 more minutes estimated. Once we're done with that, I'm going to show you how to build an app on Kavatica. And for building that app, we are going to use an existing app as a template, because it's easier when you're starting from scratch. It's easier when you have a template to work with right at the get-go. Doing everything from scratch might take a little more in-depth understanding of the whole process of app development, CWL file writing, and the command specifications and the rules of writing a CWL file. It might take a little more in-depth understanding of that, which would take us beyond 2 hours. That's why we'll start with a template. So while this app is running, let me go ahead and help you get started with that. Let me bring that up. Just like we had installed the SB pack earlier, we would need to do the unpack command. Let me find that command and then paste it in the chat. I'm going to edit that command appropriately from my template version of it, and then show it to you on the command line. I'm going to again get the entire app name from here. We are going to go in the apps tab. We're going to go to the app that we're looking at over here, and we are going to get the whole URL of that. And I'm going to make some edits to that URL just to get the parts that I'm interested in. And then I'm going to show you exactly what to do with that. All right. So in the chat is the command that I will be using to show you exactly how to copy an app along with all its dependencies and files locally. And you see here the file that we attached here to this single cell RNA-seq analysis task. It has ran successfully in 3 minutes and it has cost us 3 cents. Let's go ahead and look at the result files of that. What it has produced is essentially the report, the entire report in an HTML version of what understanding we were able to extract out of that single cell analysis. Let's look at this report. This report was able to find out the number of features that were captured in those that data object and how many of them detected features. These were false and these were true. These ones were true. And it also shows us the QC metrics across all the cells that were available for this single cell object. And it shows us this scatter plot of the PCA analysis done on this. And it also shows us the genes that are highly variable across the entire set of genes. These ones are the highly variable ones. And here is another plot of the variance of the log expression. And here is the clustering that was performed using single cell RNA-seq analysis for this single cell experiment data object. This is the clustering, and it was able to capture these 10 different clusters. And here is another, this visualization is a table where we can look at the particular gene name and the p-value and the fold change and the adjusted p-value and the cluster that it belongs to. So the ones that are in the same cluster, they are more related to each other. Their expression profiles are more similar or impacted by each other than the ones in different clusters. That is the kind of report a successful execution of that app will generate. Now let's use this same app and copy it, download it locally to see what it looks like, and then make edits to it. I have added that command, SB pull command, onto the chat. Essentially, the template is SB pull Cavatica, the username followed by the project name followed by the app tab and the app name that you want to download into Cavatica. I'm going to use this particular command on my terminal here. And I'm going to look into the current working directory where I will, let's make a new directory, CWL files, CD CWL files, and here we'll pull the files and we'll call it something intuitive, scRNA seq.cwl. This is not the app name, but I'm just trying to make it short and still informative. And we are going to unpack that whole app locally. And here I will do this is a subdirectory of the path where I was able to earlier download these files. Let's do an sbpack -h. This is just a help. And yeah, it should have worked. I have the Cavatica profile set up. In this command that you see here, Okay, local path. I think I found it. Yeah. The problem was this local path. It was going to copy it here. So then I think I can copy it in the downloads. Rowan, do you know if there's anything I'm missing here? I have the authorization profile, author profile set up, the project name.
Let
I
me
think
take a look. Sorry. I'm trying to see exactly what the issue is.
there are no hyphens, there are no capital cases here.
Um,
No, the CWF file is the wrapper of that entire app, and I'm trying to use a name, an intuitive name locally that I can download and unpack that entire wrapper, the CW wrapper inside when I unpack the app locally. That's what I'm trying to do.
yeah, it's saying invalid path parameter value. So
Is it
I'm
the
trying to figure out, I think it
path
might
or-
be the project path, or I mean, it's either the project path or where the file's located, but I'm willing to bet it's the project path because that can be.
user project name, workflow name? I got it from, okay, multi-sample Surat. Is that not the project name? Let's go back to the browser here and go to apps and get the correct path for the app.
You can also check for this on the terminal window that you were using by doing SB space projects, I think list.
Okay, let's SB projects list and then get the entire app name. Let's do that.
And that'll tell you the exact project name.
And can I do something similar for the apps?
I believe so. I'm trying to remember exactly what the command is for the apps. You can also type in SB help or SB to see the full list. Give me just a second. I can look at it myself.
Apps, it is here.
Okay.
So it does do that. Okay. So now from this entire list of apps that I have access to, I'm going to copy. There was a 0.
It's always something silly like that. So that's why I always say to check this.
Yes, yes. And no matter how many times you rehearse this, you forget one little thing and then it stops working.
Of course, yeah. That's bioinformatics.
All right, so let me get that command set up again.
I don't know if your terminal window works the same way, but you should be able to push the up arrow to get previous commands.
Yeah, it works that way, but I'm just trying to edit everything and then
Okay.
and then the CWL and then unpack. Now I'm going to copy it to the terminal so that everybody can see. And yeah, here we are. I'm going to call it crnaseek.cwl, and I'm going to unpack. I think now that the entire path to the app is looking correct, it should work. See, it was that simple. So that's what happens with bioinformatics when you work with a lot of command line. And I'm sure that when you all work with developing your tools and scripts locally, I'm sure you've seen these kind of mishaps happen to you many times. So now I was here, power up to, okay. I was otherwise working on a different folder, but never mind. Now that I have these data files, I'm going to open one of them and show it to you. Give me just one moment. It is opening up in an application called Rabbits Composer that I use locally. And when you download the entire CWL file, this is what it looks like in terms of the code. And you can also look at the same kind of code here. If you were to edit the app, you can look at the same code here, and you can look at it visually in terms of like a flow chart kind of diagram over here. And you can use this space also to make some changes in the settings. Like for example, you can double click on something and edit the parameter values or the default values, or any other app parameters that you know exist or you want to add to it. You can do it by drawing something here as well. But if, for example, if you had your own tool that you were trying to wrap up into a CWL, I'm going to demonstrate how to just use this existing template, a piece of code, edit it to show where you can just plug in your own tool. So this is the documentation that we see on the home page, like over here. If you were to go back to the app, this is the documentation, the description and everything that you see here. That's what's captured inside this documentation. Okay. So for the sake of some experimentation, I'm going to remove some of this documentation here. And let's see, it didn't have like quotes or anything. Okay. I'm just going to add a line. Power up. Demo. I just added one line, and I'm going to change the version number from 4.1410 to 10_SS, because those are my initials. And that's the label for this app that we have edited. And then other than the changes to the documentation that you can add, you can change the parameters here. These are the different types of inputs. The input type, where we had selected single cell experiment, that is one of the required input values. And there are other data types here. So you can, if you needed, you could, for example, let's remove one of those labels just for demonstration purposes. Genome build ensemble version, memory per job. Let's just remove this. And we're not going to run the new app that we're going to deploy, so it's fine. We'll just remove some of these inputs just to show you how we can edit this CWL file to make it more useful for you. And then there's the documentation for the normalization method. And then there are other input things and other parameters that are added here, which you can all edit and remove, and then get to the R script here, which is, let's search for that. So when I did that unpack, if you see here, it downloaded this parent CWL file, which is like the parent wrapper, and it downloaded all these individual CWL files also, which represent all the different individual steps of the CWL files. Out of this, I'm going to open one of these CWL files, let's say this one, which is responsible for loading some of the CWL data, loading some of the R scripts that will work with the data files and process the single cell experiment, single cell analysis. And this is it. So yes, I was looking in the wrong place, but yeah, we have it now. This is the file. And here, if you see, I'm going to look for the word load data, because I know that it exists. And after that, it's going to basically call some R script. So this is inside R script. And the script is called load multiple data sets and set metadata.r. And this is the name of the script. So if you had an R script that we're working with, you could have and it could also exist. You would have to deploy the app and your scripts, if they're on GitHub available. You would host it on GitHub and also have a Docker image of that available on Docker Hub, and link that to your Kavatica app. But essentially, this is the name of the script. And what you see here under the entry label, everything is the R script. So if you needed to, you could just edit all of this. And what I'm going to do here is that I know that we are working with single cell experiment type data. So I'm going to add a message here as a part of our effort to edit the app into something that can prove that we edited this app, we deployed this app, and this is our new app. So let's call, let's use a new error new message. Yay. Changes deployed. This is what we're going to do, and we're going to save this. Now that we have saved this, we are going to also save, and I will now, this is the file we made changes to, and we are going to again deploy this Kavatika app into a new app. Publish the Kavatika app, the project. I'm going to bring a new command onto the screen, and give me just one moment. I will edit that with the proper path to everything. Power up to, and it's called, the file is called the multi-step Surat. Rename, Control C, and here Control V. All right, so did we make the changes to the documentation? Give me one moment. Let me just verify that we have made those changes in the documentation earlier. This is a Power Up demo. Yes, we have those changes reflected here. So when I deploy this, it's going to work hopefully as expected. Let me bring up the command, and I'll paste it in the chat as well. For publishing the app, this is the command that we would work with. And let's go back to the terminal. We are here. We have made changes to the current working directory in the documentation, ls-lth, Power Up 2. We have made changes to both these files. Now let's deploy this on Kavatika. And the app name looks fine. The path looks fine. I've changed the app name to a different version. Let's publish. Incorrect path for app ID. Incorrect path for app ID.
And you're supposed to upload this CWL file into the apps portion of your project, is that correct?
Yes, that's how it works. The upload of an app goes directly into the apps tab. You just have to specify the, okay, wait, yeah, that's the part. I think I didn't have that here. I think I need to remove the apps part of it. Thank you for catching that, Rowan. You're the best.
No problem.
See?
Okay, good. That was a guess, so.
Thank you, Rowan. Now let's go to the current project and show you the newly deployed app that we have set up, and see if the changes that we made were reflected. Yes, as you see, I changed the name of the label of the app, which is visible. We removed the description, which has happened. And if we also were to edit the app, I had added some comments to it in the code. Let's find the word single cell experiment, because the changes I made were near that. Let's see if that code change, but we were able to deploy that. I'm not able to find it, and I want to take just a couple of minutes to see if I can show you how to do some debugging here. So one of the ways for debugging is to download the app. I mean, if you had an app already that you have published, then you can still add some error messages, error handling messages in your R script to track where the errors may be stemming from, and then see what changes need to be made in the R script, or in some label names, or in some parametric values. And if you're not able to do, if you're not very hands-on with that, then we have some task failures that we looked into earlier. So we can go to those failed tasks. We can, for just a head start, we can look into this error message here and look into the details of that. And here, for example, it shows us that the format converter was failing with an exit code 1. Now, exit code 1, if I remember correctly, is like a memory error, I think. But you would also look into the stats of that, stats and logs of that. And also Kavatika documentation provides additional help on, okay, it was the argument, unknown argument. Okay, so somehow the file that I had attached here was not read correctly. So maybe the process of file copying that did not successfully happen or something like that. But here is where you will see in the job error log, you will see the error messages where you can start with. And similarly, I have also had like a lot of other tasks that I worked on when I changed the apps to have enough error messages that I was dealing with. So I know exactly what is my preferred mode of action when I want to debug something. If I want to download the app, work on the R script locally, and then republish the app using command line, or if I want to make changes here in the app. And of course, you could make those changes here as well when you go to the app and you see the code for some app. You have the option of making some changes here in the code itself. And if you do that, you can just save it or just save and run the app again. And if you do that, the revision of that app will change. Right now it's 0 because I just published this app. So this is the initial version, but it can change to and you can also track those versions. You can go back to version 1 or 2 or 3, whatever you need. That's what happens. So yeah, those are my 2 favorite methods for debugging. But if you're still not able to find out what's wrong, which has happened to me in the past, you can go to the task that has failed and you can click on get support and open a ticket for Kavatika support team. From here, just write a message for them and submit the request, or the same thing like yesterday, you can use the blue question mark. And this will help the support team also track this particular app tasks settings like logs, stats, costs, the input files, and everything. So you can just add your error message here and send it to the support member. So this is also another way of finding help when you're working with something and you're not able to solve things on your own. So it's a great way to get started, whether you're working in a team or on your own. All right, so let's go back to what we've done today, what we've recap of what we've done. And let me check the new messages here. No new messages. And I know that we've covered a lot of material today, conducted a lot of analysis. We ran through 3 different apps today. One for extracting the sequence reads from the metadata file using SRA for HubMap, whatever of the HubMap data may be hosted on the SRA. We have also looked at pre-built Kavatika apps where we can convert from one data object type to another for single-cell RNA-seq data. And we also successfully ran a single-cell RNA-seq analysis and visualized the data visualize the results for that analysis. Okay, so essentially we have learned exactly what single-cell RNA-seq analysis is, the basics of it. We imported the data from HubMap via SRA and also directly via HubMap. We looked into some pre-existing apps on Kavatika, and we also unpacked the existing app to look into what the CWL file, the command line common workflow language wrapper looks like, and edited that to create a new app, and we deployed that onto Kavatika. And to be able to do that, we started with setting up the 7 bridges CLI. We have a good idea of how simple the Kavatika command line commands are to unpack an app locally, and how user-friendly, developer-friendly it is to edit an app and deploy it back on Kavatika and run again. So that's what we've done today. And I know that things were a lot more complicated today. but I hope you were able to follow along. And of course, if you have any questions, please feel free to reach out to Rowan, myself, or David, especially because if you don't reach out to us via email or anything, we'll also be available for you on during the office hours that are on Tuesdays and Thursdays. And I'll give a minute to Rowan to just help you all catch up with when the office hours are and what they do in the office hours, what the Wellsera folks do during the office hours, where you can join and learn and ask for help. Rowan?
Sure, thank you. Yeah, so I put the link in the chat. It's just it takes you to a cavatica.org webpage that just kind of tells you the times. It's 10 a.m. on Tuesday. This is Eastern time, 2 p.m. Thursdays. So today we'll have one in like 2 hours or so. It's really casual. It's me and several other colleagues. We're here to answer any questions. They can be really, really simple, like how to set up an account. They can be more advanced, like some of the stuff you saw today using the API, making calls like that. So just really anything that you need help with. If we can't solve your problem, we'll direct you to support, but at least we'll have awareness of it, you know, so we can kind of like escalate it and push it and get back to you. So feel free to stop by at any time. It doesn't have to be at exactly 2 p.m., but just whenever you're free.
Absolutely. Thank you, Rowan. Thank you for that. And
you.
everybody, I hope you were able to take from this session something useful, something that you might be able to work with in the next few weeks or months. I hope that you were able to apply for pilot funds and you were granted those pilot funds to get started. And we look forward to helping you out with your research as soon as possible. Mostly that's all I had for today, but if you have questions, we can stay on for a little longer.
Hi, Sangeeta. Just had an error with the first run. I just put a note there. I don't know whom it goes to, but I'll just wait for tomorrow and probably we can talk tomorrow about it.
So the SRA metadata.
Yeah. I
Okay.
mean, from yesterday also, I had issues with the metadata. It was not clear exactly what it is expecting for the sample ID. Is it that you give any sample ID and it takes it, or it is expecting something specific there? Because I had errors, tried running thrice, it failed on that. And today too, for the metadata extraction from the SRA, it had an error when I ran the first tool. Yeah.
Sure. If you have opened a ticket, I'm sure somebody will reach out to you via an email. And of course, you can also join the office hours where we can work with you more hands-on.
Yeah. And Uma, at the beginning of tomorrow's presentation, I can go into, I'll briefly show again the metadata fields from my demo on Wednesday. But we'll do that first thing tomorrow before we have our conversation.
Okay, great. Thank you.
All right, that's all we had for today. If we don't have any more questions, maybe we're ready to close.
Sounds good. Well, thanks so much, Sangeeta, for that great presentation. Thank you, Rowan, for all of your help today and joining.